Good morning, everybody. Welcome to the June 2024 edition of IIDR RANS. This is our last one for the year. I'm Laurie Burrows. I'm the Associate Director of the IIDR. And I'm very happy today to introduce our two speakers who are going to talk to us about a very topical global problem, which is fitting because Brian is in the Netherlands and JP is in China. So we have global presence today for our speakers. So our first speaker today is Brian Hummel. He's an assistant professor in the Department of Pediatrics at McMaster, doing full-time consulting work in pediatric infectious disease at McMaster Children's Hospital. But he's currently in the Netherlands at Maastricht University, working on a master's in health professions education. And today he's joining us by Zoom to discuss an outbreak of blastomycosis in Ontario. Our second speaker is JP Xu, a professor of biology at McMaster and a member of the IIDR. His primary research broadly involves the ecology and evolutionary genetics of fungi, including mushrooms, plants, animal, and human fungal pathogens. And today he's going to be discussing his lab's research into the changing population of fungal pathogens. So this is, I think, a really important topic as climate change makes fungi closer and closer to infecting more things that they shouldn't, including people. So thank you both. I'll turn it over to Brian to start us off.
Okay, perfect. So yeah, our presentation is called Budding Issues, Blastomycosis and Fungal Pathogens on the Move. We were proud of this pun in the title. So I'm going to start by talking about a case of a blastomycosis outbreak that occurred recently in Ontario in 2021. I'll skip introductions because I've already had one. I don't have any conflicts of interest to declare for this presentation. And it was difficult to figure out how to do a land acknowledgement on a presentation where I'm presenting from the Netherlands to an audience that's mostly in the Hamilton region about patients from Constance Lake First Nation in Northern Ontario who were admitted to a hospital in Ottawa. So a lot of moving pieces there. So I decided, well, why don't I acknowledge that this case is discussed. The case discussed pertains to people who were living in the traditional and ancestral territories of the Cree, Ojibwe, and Oji Cree people. And we thank them in the Constance Lake First Nation who continue to live on those lands and care for them, along with the waters and all that is above and below. And this is their symbol there in the Constance Lake First Nation. So this is the hospital that I trained at. It's the Children's Hospital of Eastern Ontario in Ottawa. And while I was there, it was November of 2021, when we would usually start having our wards fill up with influenza and RSV and respiratory illnesses, largely caused by viruses. But instead, we didn't have any of that. We had 4 patients on our ward with pulmonary blastomycosis, which is not something you see on your wards very commonly at all, let alone a whole cohort of them. So essentially what happened in this time, so in November of 2021, there was an outbreak of blastomycosis on Constance Lake First Nation. And this is a very small community. There's only 800 people living in this community, and over 50 people were infected with this outbreak, 5 of whom unfortunately passed away. So this was a very significant and tragic event that occurred here. And just to kind of situate you and show you where this is, Constance Lake First Nation is in Northern Ontario. These are my little Google Maps images. You can see the one on the left here, there, the lake and the small community that's right beside it. And then the broader overview here on the right to show you where in Ontario this is, very far away from where we are. And when this outbreak occurred, a lot of patients had to be transferred to other centers. The closest community hospitals, larger community hospitals were in Timmins and Sudbury, but the adults who were very unwell were transferred to LHSC, and I don't know how exactly it all was decided, but we ended up in Ottawa at the time being the ones to manage any children who needed to be admitted from this outbreak. So just to give us a little bit of background, blastomycosis is caused by an organism, Blastomyces dermatitidis, which now more recently is starting to be described as a complex because there's more phylogenetic data showing us other species. But the first one identified and the one that we traditionally think was Blastomyces dermatitidis. This is a thermally dimorphic fungi, meaning it has different structures at different temperatures. So when we are at colder temperatures, less than 30 degrees, it exists in its mold form, where you see under the microscope, you can see its hyphal structures. And so that's the image I have on the left there. This is what it would usually look like in the environment. And then at higher temperatures, it exists in its yeast form, which you can see on the right. And that's what it will mostly look like in human tissues that are at a higher temperature. From an epidemiologic standpoint, the organism likes to inhabit moist, acidic, sandy soils that are near riverbeds, lakes, bodies of water. So this is an important feature of blastomycosis. It also tends to be associated with detritus, which is dead particulate organic matter. So, decaying wood around soil that is, in riverbeds and near lakes. This would be like the classic kind of environment that blastomyces would like to live in the environment. Usually infections are sporadic, but they can happen in outbreaks, and that's usually associated with human activity. So construction, excavation activities, something like this. One feature about it is that it's very difficult to isolate in the environment. And as a result of this, its ecologic niche has really been determined by epidemiologic data from outbreaks and from infections. But it's very hard to isolate in the environment. You can see here when people isolate it, they publish about it because it's so challenging to do. So a lot of our data about where blastomyces exists is from epidemiologic data as opposed to actually detecting it in the environment. So here you can see a figure here that shows the endemicity. So it's hyper-endemic to Northwest Ontario, Minnesota, and Wisconsin. These are kind of the highest prevalence regions, but that endemicity extends essentially all along the Great Lakes and the river basins of the St. Lawrence, Ohio, and Mississippi rivers. So that's why you can see it kind of extending down into the southern US, because it's following along the Mississippi River and the Ohio rivers, and why you see it extending up along the St. Lawrence there. So that the proximity to waterbeds is key with blastomycosis. And the range is expanding. So there's data from Quebec that is showing increased incidence. We have data in Ontario as well. And we're even seeing that it's spreading, moving out west to provinces like Saskatchewan. We're even seeing the incidence increasing in dogs. So humans are our main host, but also dogs get infected with this relatively commonly compared to other animals. And so you can see here in this figure from some data from veterinary medicine that the incidence in dogs has been increasing in Ontario as well. This is a recent Canadian Communicable Diseases report published in 2017. So this looks at blastomycosis incidence in Ontario over 10 years from 2006 to 2015. And at this time, it was not a reportable disease in Ontario. So this data is all from ICD codes from hospital admissions only. And they've described 586 cases over the last 10 years, mostly in the Northwest Ontario Health Unit, which you can see on the figure in the lower left there. And you can see they're showing, they're able to demonstrate an increasing incidence. in Ontario, which is a problem and is hypothesized to be related to climate change. So I looked on the Public Health Ontario to see what our kind of more recent data is, because it would appear in 2018, after this data was reported, that it became a reportable disease in Ontario. And so now we have incidence data. And so I pulled this all from PHOL, and you can even see from this figure that number of cases is on the rise. Some brief points about pathogenesis and clinical manifestations. So this is transmitted by inhalation of aerosols, spores from the environment. It's not human to human. You have to inhale it from the environment. Very rarely you can have skin inoculation, but that would not be classic. It's mostly inhalation of aerosolized spores from these rotting wood at river basins. And it does infect both immunocompetent and immunocompromised hosts. It has an incubation period around 4 to 6 weeks would be classic. 50% of people are asymptomatic. The other 50% will have clinical manifestations, and it's typically a primary infection as opposed to reactivation disease. You can have an infection with reactivation later in life that has been described, but it is classically a primary infection. And it's usually an acute or subacute pulmonary syndrome, right? So it likes to infect the lungs primarily. So your symptoms are, cough, fever, night sweats, fatigue would be classic, and you get a very patchy consolidative pneumonia classically, though it can be nodular or cavitary and look like even something like tuberculosis. And the reason it's a disease of concern is because you can have very severe manifestations too, with a diffuse pneumonitis and an ARDS, which is why you can have fairly high mortality from this. And it does cause extrapulmonary disease as well in a relatively large number of cases. In order of incidence, skin would be the main one, but also bone, genitourinary system, and brain. But that's just to give a little bit of foundation. For the cases that we saw during this outbreak, I'll just run through the 4 of them. So one of them was a 2.5-year-old male who was previously well child. He had 3 days of cough and fever, but was otherwise very well appearing, didn't need any oxygen, didn't need any work of breathing. And his chest x-ray showed this very small right parahilar opacification. So this, not a particularly impressive presentation, not a particularly impressive x-ray, even just looks like an early bacterial pneumonia, but this patient got picked up very early because we knew that they were part of this outbreak. Here's another one of the patients. This one is a 6-year-old female. She was also previously well, who presented with, again, very more acute symptoms, chest pain, fever, cough, and wasn't hypoxemic, no work of breathing, had a more impressive pneumonia on x-ray compared to our last patient. And then we have the patients who are a little bit more sick. So this is a 13-year-old female, previously well, 10 days of dry cough and headache, progressive cough, more productive, then had high fevers and was needing more respiratory supports, tachypnic, 2 liters of oxygen requirement. And as you can see here, a much more impressive pneumonia, bilateral, very patchy. This is not something that looks like you'd mistaken it for a bacterial pneumonia, a typical bacterial pneumonia. So this one's more impressive. And then this patient was the most ill, 5-year-old, previously well, who had 3 weeks of respiratory symptoms and required more respiratory supports, high flow oxygen with this patient. And you can see here a much more impressive pneumonia syndrome. It is likely that these 2 patients, had they not been picked up early in an outbreak, high pretest probability outbreak setting, they may have presented more like these patients with longer duration of symptoms and more impressive features. It's always fun to look at the microbiology when we're talking about blastomycosis. So this was a sputum culture of that last patient, the most sick one that I told you about. This is just a regular gram stain, but the burden of disease was high enough that you could pick this up on gram. So you can see here, this would be a warmer temperature, right? Because we're picking up the human specimen, the sputum from the lungs. So you can see it in its yeast form here, right in the middle there. You can see, I don't know if you can see my cursor here, but the classic description is a broad-based budding yeast. So you can see these circular yeast forms, and it is next to its, the daughter yeast is next to the mother yeast here, and they're budding off with a really broad base. They're larger in size, so 8 to 15 micrometers, and you can appreciate this this thick sort of refractile capsule as well there. So this is a very, very typical and classic features that you can pick up. This is not my gram stain. This is just a nicer image of a calciform white stain that shows you this broad-based budding feature that I am referring to. And then 1 week later, the fungal culture grew this. So this is, again, this is my own picture here of this patient. You can see now, because we're growing it in the lab under cooler temperatures, we're seeing it grow in its mold form. So you can see these hyphae, and the classic feature here that you can see is these little branches that are budding off at right angles from the main sort of hyphal stem. So we would call these conidiophores, branching off at right angles with a single terminal conidia at the end of it. And these look like lollipops. So this is what it's often described as, little lollipop-looking things. From a diagnostic standpoint, of course, you know, the typical clinical syndrome and epidemiologic history is your first step to elevate your pretest probability. But the gold standard of diagnosis for this would be culture. But of course, this takes time. For more rapid diagnostics, we have cytology, right? So looking at the direct smear. But of course, you don't always get favorable specimens, especially if the burden of disease isn't as high, and especially if you have 2-year-old children who don't want to produce a sputum. So if you do get samples, the sensitivity can be low, but if you do see the organism, it is pretty specific because of those very characteristic features. There are some ancillary tests you can do if you're running into issues with sampling. For example, we have a urinary antigen. Well, we don't have it. It's not offered in Canada, but we can send it to this lab in Indiana called Maravista Lab. And there are some sensitivity and specificity issues that I may get into briefly, but I might run out of time. But this is another ancillary test that we can do. There's also serology that's available at Public Health. They do an immunodiffusion test that, when I read about it, is not the best serologic test in this context. There is an ELISA that's better, that PHOL, I guess, doesn't want to do, because overall serology is not the most helpful test in this setting. And then molecular diagnostics are actually pretty scarce for blastomyces. So there's nothing that's offered in Canada, and there are only a few labs that offer this. I haven't been in a situation where we've attempted to utilize molecular testing yet. I was going to get into a little bit about when the urinary antigen might be helpful, but I'm conscious of not running over time, so maybe I will try and tell you briefly. Our guidelines for this are very old. They're published in 2008. So the data behind this is not up to date, but they basically are non-committal with urinary antigens. They say the role of this test should be the role that this test should play in diagnosis of blastomycosis has not been established. So they kind of are non-committal about when to look to using this test. And I had at the time, you know, looked into the literature for this a lot, and the gist of it is that the sensitivity isn't great, very variable, and the specificity is limited by cross-reactivity to other thermally dimorphic fungi. So if you get a positive urine antigen test, you don't know if this is a different thermally dimorphic fungi, for example, like histoplasmosis. And the reason for this is because there's nanoproteins in the fungal cell wall that the assay cross-reacts with. So you have to be really kind of considerate about what situation this is useful in, and really be careful with interpreting results. Obviously, if you have a direct positive smear, it's helpful. I won't kind of go through all of these scenarios, but my take on it is that if you have a direct negative smear or if you have a sample, there is still good utility in this test, particularly in a higher pretest probability setting. But I don't want to take JP's time, so I won't outline that more. So in our patients, we had the patients with higher burden of disease, as you might expect. They had positive sputums. You could see it on cytology. We grew it on culture. Didn't need urinary antigens. It was very easy. The patients who were picked up earlier and who had more mild symptoms, the patient with the larger pneumonia had a positive urine antigen, but the fourth patient, they had a negative urine antigen, and this is where it becomes hard to interpret because with a lower burden of disease, it's hard to judge if that's false negative or not. This patient was going to get treatment whether that thing was negative or positive. And treatment, I'll mention briefly, is very prolonged. So we use oral itraconazole for 6 to 12 months if the disease is mild to moderate, but patients who are more ill, like two of our patients there anyway, we do an induction therapy with IV liposomal amphotericin B. And we give this for a period of time up front and then eventually step them down to oral itraconazole and continue that for several months. So when we dug back in the history for these patients to figure out, well, where did this come from? What was the exposure? It turns out that a month before presenting, so in October, before they presented in November, there was a big bonfire that they had done as a community, and they had used this all of this wood that had been stockpiled throughout the summer, and even into years past, that they did describe as, some of that wood looked pretty waterlogged and decaying and kind of rotting wood. And so you wonder if that could have been the kind of outbreak event there. They were chopping up this wood and bringing it over to put in this fire. So that certainly could have aerosolized the organism and infected a large number of people. And of course, this First Nation is right on this lake, right? Constance Lake, that would be connected to Lake Superior. So I tried to, look into if they ended up finding an environmental source of this, of the outbreak, and I couldn't find anything published, but I had to sort of piece this story together just through reading media reports, and it would appear that Sutherland is the chief of that First Nations, and so you can see in this report, investigators searched around 200 locations. All their tests came back negative, and so this was initially sort of spearheaded by Indigenous Services Canada and this laboratory in Toronto, this private lab, Sporometrics. I don't know what testing they did on what samples, but it looks like their effort to try and identify the organism in the environment was all for naught. And as a result of this, the community remained in a state of emergency for over a year while they were trying to find the source of this outbreak. But eventually they just didn't find anything and kind of moved on. But interestingly, I remember this case is from 2021. When I was making this presentation, I found, oh, wow, like recently in February 29th of 2024, there was this article from Matawa First Nations, which is a group of First Nations that includes Constance Lake. They released this media report that said, oh, we have detected blastomyces by DNA. And I was like, that's interesting. Who did that? Like, what happened here? And learned that the Matawa First Nations Four Rivers organization, which is an environmental advocacy group, basically, partnered with Laurentian University and this organization called Up North on Climate that partners with Indigenous communities to try and solve climate change related problems using a combination of traditional Indigenous knowledge as well as scientific endeavors. and they try to solve climate change related problems. So they kind of took this on themselves, a really grassroots effort. And when I was reading their reports, they made reference to the University of Minnesota, where they were able to send something. Again, I don't know what they sent, but they sent something that was in their environment that was positive for Blastomyces DNA. And lo and behold, when I look on the University of Minnesota, or the Department of Health in Minnesota's website, they've recently offered a Blastomyces PCR. And so it sounds like they sent, I don't know, maybe wood from that pile that they still had stored somewhere, and it was positive by PCR. So I just thought that was a really cool example of a grassroots advocacy effort that ended up solving a climate change related problem for this community. So I thought that was an interesting story to share. But I will not take too much more time because I need to leave enough for JP to begin. So I will pass it on to JP.
Thank you, Brian, for then acknowledgement. I will not repeat that here. So what I'm going to describe are a few recent studies in my lab, as well as with my collaborators, on how fungal pathogens are changing very rapidly. Back in September 2022, I used to primarily publish the literature describing the fungal disease burdens in humans. So blastomyces was one of those that was included, and the global burden was estimated to be about 3,000. But this is really a very small percentage or small number compared to the global burden of fungal infections. As you can see over here, superficial infections of the skin, hair, and the nail, it's over 1 billion people. And for chronic pulmonary aspergillosis, it's several million, 3 to 4 million cases. And another high number invasive infections, high incidences were the invasive candidiasis. Those include candidemia, and they are caused by a variety of Candida species in the genus Candida. For Candida, they can also cause other diseases as well in other body sites. For example, the current vaginal candidiasis, it's hundreds of millions of cases globally. And then a month later, after I wrote that paper, this WHO's priority fungal pathogen list came out. And it included the 19 species into 3 priority pathogen group, including critical group, high priority group, and medium priority group. Blastomyces did not make the cut in this list because it's primarily a local and endemic disease around the Great Lakes. But certainly it's becoming more important. So over here, we have worked with all 4 species in the critical group, as well as other species in the high and the medium group. But today I'm only going to introduce you our recent studies in 2 of those 4 critical group fungal pathogens, Candida auris and Aspergillus fumigatus. And I'm going to introduce them to illustrate 2 major points, linking basic biology to clinical importance. So first, there's a story on Aspergillus fumigatus. The take-home message that I want you to get from this little story is the rapid emergence and spread of antifungal resistance in the global population of this species. So A. fumigatus is a very common mold in our environment. It's in the air, in the soil, in the water, and in nature, in the environment, it primarily acts as a saprophyte, a nutrient recycler of organic matter. Every day we inhale hundreds of spores of this species. And if you work with organic matter all the time, like gardening, etc., you probably inhale more spores of this than a few hundred, perhaps up to millions of spores. And the spores of this species can be isolated from air sample collected from stratosphere over 30 kilometers above sea level, and has also been isolated from the International Space Station. Two features of this species make it a very potent pathogen. One is that it's thermotolerant, can easily grow in a variety of environments, very easy in a human body temperature, unlike many other fungi that they cannot grow at 37 degrees or above 37. The other one is the production of very abundant and small spores. A single spore landed on a petri dish. After a few days, after germination and growth of a few days, it can produce billions of spores like this over here. And each of those spores is hydrophobic in that they can easily pass through some of our airway. But for most of us, it's typically not a problem. Our immune system, our respiratory system can clear the spores of this species. Our work on Aphiomegatus started in 2012, just over 10 years ago. I was invited by colleagues from the Netherlands and India to analyze the genotypes that they obtained from environmental and clinical samples of this species from across India. In that analysis, we identified that there was a very large clonal genotype distributed in many different ecological niches throughout India, stretching hundreds of kilometers. And all of those strands have the same genotype at 9 microsatellite loci. Those are highly polymorphic loci in the genome. What's striking about this cluster is that they are resistant to multiple triazole agents, including voriconazole, itraconazole, the two common triazole drugs that are used for treating patients infected by Aphiomegatus. Based on this and other reports by other scientists from other geographic areas, we proposed that the emergence of this cluster, as well as potentially other azole-resistant strains, were likely due to the use of triazole fungicides in agriculture. And many of those agricultural fungicides have the identical mechanism of action as clinical triazoles. And quite often, the strains that are resistant to agricultural fungicides are also resistant to medical triazoles. And globally, fungicide production and application are increasing year over year. And this is likely going to drive the expansion of triazole-resistant pathogens. To test for this hypothesis, we choose a specific ecological niche, the greenhouses. It was chosen because greenhouses are becoming increasingly important space for growing a variety of vegetables, ornamental plants, as well as other kind of food-related items. And this is from China, the greenhouse space expansion. And for greenhouses, they also have quite different ecological niches than natural environments, in that they typically have a higher temperature than the surrounding environment. The crop growth is typically much more intensive than outside the environment. Usually it's year round. And they also are isolated from other greenhouses. Therefore, they create a unique ecological niche that potentially could facilitate the development of drug resistance. And in the growth of many of the crops, vegetables, ornamental plants, there's also heavy use of agricultural fungicides. So we conducted a series of studies to try to identify the potential hotspots for triazole resistance in Aphiomegatus. And those were reported in 3 studies. One was in 2021, where we found high prevalence of triazole-resistant strains of this species from the soil of greenhouses. Specifically, we isolated over 200 strains from 9 greenhouses. And we found that 80% of the isolates were resistant to at least one of the triazole endofungals. And over 30% were resistant to at least 2 of those endofungals. To examine whether this is a unique event, whether greenhouses are unique, we also analyzed natural environments, including agricultural fields, national forests, and water bodies, including rivers and lakes. In those environments, we analyzed a total of over 400 strains, and we found 16%, about 16% of those isolates were resistant to 2 clinical triazoles, itraconazole and or voriconazole. So the frequency is lower than the greenhouses, supporting probably greenhouses is a big hotspot. But still, we could not exclude natural environments such as not natural, human impacted, such as agricultural fields as a source. And then to further examine how widely spread the triazole-resistant population might be, we looked at the 3 parallel river regions that are very sparsely populated, very remote from many of the civilizations. And in those environments, we still found about 7% of the isolates were resistant to either itraconazole or voriconazole. And we also tested the potential resistance mechanisms of those strains that showed high MIC. Only about 30% of the isolates, resistant isolates, had the commonly identified mutation in other parts of the world, including a repeat in the promoter region called the TR34, and a specific amino acid substitution at position 98 called L98H. So about 70% of the isolates have non-canonical resistance mechanisms that involving genes in other parts of the genome, not the target of triazole. So that's Aspergillus fumigatus, in that we identified at least a potential, or likely a major hotspot for triazole resistance development and then spreading to other parts of the environment. The second story that I'm going to tell you is about the Candida auris. Now, some of those stories of our work on Candida auris have been reported by IIDR as well as McMaster News. Here, I'm only going to focus on one of the basic questions that we are interested in, which is the origin and the mode of reproduction of this species. Typically, we think of the emergence of new diseases as some kind of very major change in its genome. So one of the hypotheses we want to look at is whether the emergence of Candida auris was due to sexual recombination, generating novel genotypes that suddenly become very virulent or resistant to environmental stress. The main message from this story is that this origin of Candida auris is primarily, almost exclusively, due to clonal mutation, clonal expansion, and mutation, with very little evidence for recombination. But there's recombination. So Candida auris was first reported in 2009, and now it's found in over 60 countries and caused thousands of deaths along the way. The major characteristics of this pathogen include its ability to infect multiple body sites in humans, and is resistant to multiple endofungals belonging to all the major endofungal drug classes. In addition, it's also tolerant to multiple stresses, including high intolerance to high osmotic pressure and to high temperature. They can grow relatively easily at temperature above at 42 degrees. And so those abilities make it highly prevalent and persistent in clinical settings, quite often using a variety of disinfectants you still cannot get rid of them in the clinical environment. So it's a major cause of nosocomial infection. Genomic analysis of Candida auris have identified 6 clades. Among the clades, there are thousands to tens of thousands of SNPs across the genome. But within individual clade, there's very few genetic differences at the whole genome level, from a few SNPs to maybe a few hundred to a couple thousand SNPs. The 6 clades belong to two mating types, like sexes in animals. The mating types are called A and alpha. Clade 1, 5, and 6 has the mating type of A, and clade 2, 3, and 6 mating type alpha. Sorry, this is for A is 1, 4, and 5. Alpha it's 2, 3, and 6. To examine whether the emergence of this species evolved a recombination, we took a population genomic approach. When we were studying this research, that was at the end of 2001, there were about 500 published Candida auris genomes. At that time, almost every isolated Candida auris strain was sequenced at the whole genome level. And then we analyzed the data, we submitted it, and submitted the manuscript. And then about 2 months later, which was in April 2022, the number of genomes already doubled from about 500 to over 1,000. So we had to reanalyze all the genome again, downloaded all the genomes and reanalyzed all of them. This also illustrates the importance of this species in the medical community. So we retrieved the whole genome sequences of all the 1,000, at that time, 1,037 published Candida auris genomes, called the SNPs for both the nuclear and mitochondrial genomes for each of the strains. And we called the SNPs using the total population as well as the clade-specific population. And then we conducted 2 tests. When I say we, it's actually my PhD student who did all the computational work. Did 2 tests. One is called phylogenetic incompatibility, also called the 4-gamete test. Another is called the linkage disequilibrium. And here I'm going to provide 3 tables summarizing our main results. The first result is evidence for infrequent recombination in both the nuclear genome and the mitochondrial genome in the total population. So typically, we don't think of recombination in the mitochondrial genome, but for this species, we find evidence for recombination in both the nuclear and mitochondria. But the rate of recombination is very low. Only about 2.8 and 2.5% of the SNP pairs were showing evidence of recombination. So that's in the total population, about 2.5%, just over 2.5% in the total population. Now then we did nuclear genome for each of the individual clades. We also found evidence of recombination, but it's very low, even lower than the total sample. At that time, there were 5 data for 5 clades. Clades were only reported last August. Clade 5 only had 5 strands, so we could not include clade 5 in the analysis. And all the clade 5 strands were from Iran. Now, for the 4 remaining clades, clade 1, 2, 3, and 4, we had variable number of strands. And the frequency of SNP pairs showing evidence of recombination is much lower than in the total sample. So this is the nuclear genome. And the conclusion over here is that less frequent recombination in the nuclear genome in the clade-specific samples than in the total population. And we then analyzed the mitochondrial genome to see whether there's any evidence of recombination within individual clades. Among the 4 clades that we analyzed, clade 2 and 4, the whole mitochondrial genomes were monomorphic, consistent with their very recent ancestry, recent emergence of the samples that we have in the gene bank. For the remaining 2 clades, clade 1 and clade 3, we found no evidence of recombination in clade 3, but we did find evidence of mitochondrial recombination in clade 1. So together, the results suggest that there's more recombination before the split of the clades than after. And then there's recombination in both the nuclear and the mitochondrial genome. We typically don't think of mitochondria recombined in animals. And indeed, there's very little support for mitochondrial recombination in the animal literature. But in fungi, we found evidence of recombination not only in this species, but also in several other species. Okay, so that's the second case. The next one I'm going to introduce is rapid evolution of an animal fungal pathogen. Many of you probably have heard of white nose syndrome in bats. And this is caused by a cyclophilic fungus called Pseudogymnal ascus distractans. This fungus and the disease is so far the most deadly fungal pathogen to mammals in the recorded history. Over the last 18 years, it's killed millions, probably tens of millions of bats in North America. Our interest in this fungal pathogen started in 2012. So this fungal pathogen was first discovered in 2006 near Albany, in a cave near Albany, New York. Since then, it has expanded north, south, east, and west. Now it's found in all 10 Canadian provinces and in most of the US states. The speed of spread is hundreds of kilometers each year. During this spread, mutations have accumulated. Those mutations are affecting the phenotype. When we first looked at this fungus was in 2012, was started by undergraduate thesis student, Jordan Kankert. What we found at that time was that strains from different geographic areas had very different morphology. However, at 8 loci that we sequenced, they were all monomorphic. So consistent with a single introduction of this species into North America from Europe. Also consistent with the Caven log data. Somebody from Germany probably brought it over. So the phenotypic variation can be very easily seen on some of the colony morphological features. It's shown over here. And this kind of morphological feature change actually have geographic gradients. Shown over here are 2 of our studies published in 2018 and 2022 in AEM and the Journal of Fungi. The x-axis showing the distance of the isolate from the center of origin, which is near Albany, New York. When the distance goes further away from the center of origin, we found some positive correlation in the phenotypic traits. For example, the colony growth, colony size, and the pigment production showed a positive trend. And the west-east direction also showed some statistically significant trend. Now, we do acknowledge that there's a lot of noise in the data, but with that kind of distribution and geographic variation, the signal is still there, showing geographic gradient of the phenotypic change in colony morphology. Dozens of genomes have been sequenced for this fungal pathogen. At the top is a strain from China, Jilin. And then over here, actually, I should have had the cursor on. Sorry about that. Oops, sorry. No, not that one. I need to. Here we go. Okay. Over here, it's primarily from Europe. And then the one closest to the North American strains, which were all here, is from Czech Republic. So over here, at the bottom ones are those that were isolated from different parts of North America. The isolates shown in green color over here are from the States, in blue are from Canada. So since its introduction in 2006, this pathogen has diverged into two different major clades. One clade over here, another clade over here, primarily through mutation accumulation. And we certainly see evidence of isolates going back and forth, genetic relationships between isolates going back and forth between Canada and the US for both clades. Most of the isolates that are not highlighted, the isolates over here and the isolates at the bottom are not highlighted, were isolated from before 2016. And since 2016, and all those were based on pure culture, since 2016, nobody had submitted any more sequence. And then just recently, we did a metagenome analysis of guano and wind swabs from Western Canada, including Alberta, BC, and Saskatchewan. Again, in those last few years, we found significant accumulation of mutations among isolates within Canada. So this species is changing very, very rapidly. And some of those genetic changes probably contributed to its adaptation and spread to very different geographic, ecological niches and geographic areas across North America. So this is the third example of how fungal pathogens are changing very rapidly. The last one I'm going to introduce is a plant fungal pathogen. And again, I'm going to try to demonstrate that greenhouses can facilitate rapid evolution of soil fungi. Again, let's go back to greenhouses. The fungal greenhouses is a very unique environment in that it has high temperature, at high intensity agriculture. This high temperature potentially could mimic climate change in different areas. And high intensity agriculture also implies frequent use of agricultural fungicides to keep the productivity, to maintain productivity. And physical isolation here means that it could potentially act as a barrier for gene dispersal, for gene flow among greenhouses, even within the same geographic area. And together, we suspected that this kind of structure that is completely artificial, created by humans, could facilitate pathogen evolution, as well as evolution of other soil microorganisms, including soil fungi. So the fungal species that we choose was called Alternaria alternata. This fungus can infect over 380 economic plant species, including most of the cereals, potatoes, tomatoes, cucumber, and almost all the fruits, oranges, apples, etc. All the major economic plants could be infected by this fungus and can reduce productivity by up to 80% for some of those crops in certain areas. And this species also shows similar kind of resistance that we are seeing for some of the human fungal pathogens. In addition to causing economic loss for agriculture, this species is also a very common allergen, and it can also cause human diseases as well. For plants, it primarily causes leaf spots, black spots. For fruits, it's soft rot. For humans, It can cause diseases in a variety of body sites, including onychomycosis, eye infection, and skin infection, subcutaneous infection. So it's one of those rare species that can cause diseases in both plants and in humans, as well as in some of the other animals. So the first study we did was published in 2022, where we isolated soil isolates of this species from the same 9 greenhouses in southwestern China. Here, we isolated over 200 isolates, genotyped them using 10 microsatellite markers, and determined their susceptibility to 4 triazoles. The genetic analysis of those 9 greenhouse populations of this species revealed that different greenhouses have genetically distinct populations. For example, shown over here, you have population 8, showing very different genetic composition, very different gene frequency and genotypes as those found in the remaining 8 greenhouses. They showed very high resistance as well to 2 clinical triazoles over 25%, and 2 agricultural triazoles, one of them is over 50% of resistance, with MIC greater than 8 microgram per ml. Prevalent drug resistance. So how does this greenhouse isolation create the genetic differentiation and evolution compared to long-distance geographic isolation? To test this, we isolated this soil isolates of this species from 7 greenhouses in another city, which is located about 2,000 kilometers away. We did similar kind of analysis for genotype and drug susceptibility. Here shows the genetic analysis of this 16 greenhouses. The result shows that greenhouse isolation from within the same area, so this 9 greenhouses from southwestern China was located in 5 kilometers away, in 5-kilometer range, and the same for over here. What you see here is that the greenhouses from the same area may show the same or even greater genetic differentiation than the populations between those populations located over 2,000 kilometers away from each other. And we also found very prevalent drug resistance. Shown over here, you have one of them, agricultural fungicide, almost 80% of the isolates were resistant to this tebuconazole. So that was just published less than a month ago. So to summarize, greenhouses can facilitate the pathogen evolution, and this is primarily due to human activities. And those can lead to high prevalent drug resistance and the genetic differentiation among greenhouse populations. A single accidental introduction, such as the white nose syndrome fungus, can have very devastating consequences, causing billions of losses to agriculture due to the increased pest population to agriculture crops. We also show the evidence of frequent gene flow among microbial populations from diverse taxa. And for Candida auris, this asexual reproduction and mutation led to the emergence of divergent clades within this species. And together, this results suggest very close linkage between agriculture and human health in both the pathogen evolution in drug resistance and how those traits could be passed on to human populations. So I stop here. Thank you very much for your attention. I'm sorry I ran a bit over time.
Thank you very much. Fortunately, Brian has been on top of it and answered most of the questions for his section in the chat. So I just have one quick question for you before we sign off, and that is, because of this intensive greenhouse environment, has there been any documented human transmission of some of these fungal pathogens? Any greenhouse workers been infected?
Yes, so we're analyzing the data right now within southwestern China. We have collected samples from the air, from outside of the greenhouses, from hospitals, from markets. There are few shared genotypes, but most of the genotypes are different. And that's very similar to the greenhouses where we found very few repeated genotypes. Most of the strains have distinct genotype, but the alleles are very similar, are very identical. So we could track that for some of those isolates from humans, they could be easily generated by recombination between isolates from greenhouses and from other areas.
Yeah, it's very scary to hear that you're finding aspergillus that is resistant to most of the antifungals. Okay, because we're short on time, I'm just gonna close by thanking both of you for your great presentations. And as usual, hearing about fungi, I find a little bit terrifying, but it's useful to have the knowledge. Have a great day, and I will see you, everybody, back in the fall.
Thank you very much. Yeah. Thank you, Brian,
so
for
much.
sharing the podium.
Thank you.
Thank you as well.